This section of the tissue level of organization chapter is going to be focusing on the connective tissues. The connective tissues present a little bit of a problem in that they're not really all connective tissues. They were discovered based on the actual connective tissues initially, and then these other tissues came to be understood to be part of the same family, while they don't exactly connect things together. Now, a lot of sources will explain that away by saying that they connect things in a very metaphorical sense, but it's different than what connective tissue was originally named for. What really ties all of those tissues together is that they all come from the same embryonic tissue called mesenchyme. Now, the connective tissues proper, which we'll talk about in a little bit more depth in a little bit, are the ones that actually connect things together. And then cartilage, bone, and the liquid tissue, blood, aren't really holding things together. We associate them with connections, whether it's joints or bones or the circulatory system, but they aren't really connecting things together in the same sense as like a tendon would. But all of these tissues come from the same source, which is this embryonic tissue called mesenchyme, and that's really the definition of what makes things a connective tissue, along with the properties that they share, which I'll talk about in a little while. This table from the book classifies the tissues similar to what I just showed a little bit ago. The connective tissues proper are the ones that actually do the connecting to things. There's a loose version and a dense version, and there are subtypes of each of those. Then the book uses the terminology supportive connective tissues for cartilage and bone, because again, they don't actually connect things together. We find cartilage at joints, and so that seems like a place where there's connection, but actually what's connecting bones together at joints are the connective tissues proper, mostly dense irregular. And then the fluid connective tissues listed here are just a class unto themselves because they are mostly water. Now, when I get to talking about the fluid connective tissues, I'm going to concentrate on blood and explain why I don't think lymph fits as a connective tissue itself. But more on that later. The first type of connective tissue to look at more closely is the loose connective tissues. Now, there are three types here. They're areolar, reticular, and adipose. An important part of what makes connective tissues is really based on how their extracellular space is really important to their characteristics. All connective tissues, even the ones that don't actually connect things together, share in having an extracellular space, which is often referred to as a ground substance or sometimes even a matrix, with particular cells suspended in that and fibrous proteins that give it some functional properties. And a great example of that is areolar connective tissue, which we'll start by looking at that more directly. Areolar connective tissue is a great example of what your basic connective tissue is. We find it all throughout the body. There's no one organ that's a better example of it for any other. It's found everywhere. In the schematic representation of the tissue that we see here, there are small and large fibrous proteins. The large ones are collagen fibers, which we find in most every connective tissue. And then the smaller ones, the green ones in this picture, are elastic fibers, and the darker ones that look like a spider web at the top are reticular fibers. Reticular fibers are actually a variation on collagen fibers. They're just thinner and more mesh-like. Reticular actually means network. And then in the spaces between these fibrous proteins, we find various types of cells. The main type of cell is the fibroblast. The fact that the name of that ends with blast refers to the fact that it is a stem cell type of cell. And fibro is referring to the fact that it's responsible for making most of these fibers, like collagen. The other cells that we see here, there are adipocytes, which are fat cells. A macrophage is a type of white blood cell. And then they point out a mesenchymal cell, which is really a remnant of the embryonic tissue. Mesenchymal cells and fibroblasts are similar cells. There are a little structural differences here and there, but they're kind of the same thing. And the mesenchymal cell here could easily become a fibroblast to do its job. But mesenchymal cells also have the ability to become adipocytes or some other types of cells that are not mentioned here. An important type of cell in loose connective tissues is the mast cell. Now, it's part of the immune response. It's responsible for initiating inflammation by releasing histamine as a signal. You're probably familiar with this because you might have taken antihistamines for an allergic reaction. And antihistamines, in fact, target mast cells and keep them from releasing that signal, which is going to keep inflammation down, which is going to take care of a runny nose or watery eyes or puffiness or whatever. Those are all symptoms of inflammation. This is reticular tissue, another type of loose connective tissue. And it's similar to areolar connective tissue in that it has space between the cells that's filled with fibrous proteins. The reason it's called reticular tissue is because the main type of fibrous protein found there is reticular fibers. Now, they look a little bit different in the representation here, but they're essentially the same as what was depicted in the areolar connective tissue. It's a network of fibers, a mesh, giving the tissue some support. Now, reticular tissue is best known for being the tissue in lymphatic organs. So lymph nodes, the tonsils, the appendix, red bone marrow, all of those types of tissue are reticular tissue. There's some variation from organ to organ exactly what it looks like, but they're all reticular tissue. In this field of view that we see in the micrograph, you can see some closely packed cells and a little bit of space here and there that's clear of cells. And that's something that you'd expect to see for reticular tissue. Within a lymph node, for instance, there are cell-free areas where fluid can accumulate, and then cell-dense areas that are important for the function of that particular type of organ. The third type of loose connective tissue is adipose tissue. Now, under the microscope, it can look a lot like the other types, and maybe even seemingly looser or clearer. But really what it is it's a collection of adipocytes. And adipose fat is a lipid and does not stain very well because it's not a charged particle. And so we don't really see the fat. All we see is the cytoplasm of those adipocytes. That's a thin little outside covering to the cell and the occasional nucleus. So it seems to have a lot of extra space in it, like areolar and reticular tissue. But in fact, the cells are rather densely packed together. However, there is some extra space. There could be blood vessels moving through and those sorts of things. But what really does associate it with the loose connective tissues is that it has a very similar consistency. Now, the loose connective tissues are best defined as having a gel-like consistency. And as collagen is sort of the main fibrous protein found in those tissues, that makes a bit of sense. Jello is actually a collagen solution that's allowed to set and gelatinize. Gelatin is really just the name for collagen after it's been rendered or processed and removed from tissue. It's just hard to sell a box of rendered collagen-O, so it's called gelatin. That's a little silly kind of way explaining it. That's not really where gelatin comes from name-wise, or jello, but that's a fair description of the molecular basis of jello, which is very similar. Now, fat is similar to that also. At room temperature, it's going to be a more solidified consistency, which is what you might have had experience with if you've dealt with fat on raw meat that you're preparing for dinner or something like that. But in your body, at body temperature, it's going to be a softer tissue and more in keeping with the gel-like consistency of the other two. Now we come to the dense connective tissues of the connective tissues proper. Now, in the table in the book earlier, they misrepresented an aspect of this, which is that there's actually three types. There's dense regular connective tissue, dense irregular connective tissue, and dense elastic connective tissue. What defines all of these is that they're mostly made of collagen. The ground substance has very little empty space in it. It's just collagen fibers all packed together. And how those collagen fibers are packed together gives a good idea of the differences in the tissue. This figure shows dense regular and dense irregular connective tissues side by side. And really this is a good way to consider them rather than separating them out, because they're essentially the same thing, which is densely packed collagen fibers with the occasional fibroblast there to help maintain those fibers. The difference is, and this is the root of why they have different names, how the collagen fibers are arranged. Regular is parallel fibers, and irregular is fibers just all sorts of different directions and angles and crossing over each other. The reason for that, both of these types of tissue serve a similar purpose, which is to absorb tensile stretch. Regular connective tissue is found in places where it's a very linear tension, and irregular is when tension can be applied in random directions, all different directions. We see regular connective tissue in tendons where it's point A to point B, from the muscle to the bone, and all tension is expressed along a single line. And then irregular is found in places like joint capsules and the deeper parts of the skin, where tension can be applied in a lot of different directions depending on what that tissue is doing at the time. This is a micrograph representing the elastic dense connective tissue, and this is a special version probably of the dense regular connective tissue, where there's a high proportion of elastic fibers mixed in with the collagen fibers. Now, in this micrograph, we see that because it's been stained to highlight the elastic tissues. So the darker threads that we see in with the collagen here is the elastic tissue. This happens to be the wall of the aorta, which is a perfect place to see that. As the heart beats, it pumps a bolus of blood on into the aorta, and so the wall of the aorta can stretch very quickly to accommodate that, and then the elastic fibers will return the wall of the aorta back to its original shape, which will help to push the blood on through circulation. We also see dense elastic tissue in the wall of the lungs. As we inhale, the lungs stretch out because of muscle contractions pulling it out, and then the elastic fibers there will help to pull everything back to its original shape, and that recoil of the elastic fibers helps to push air out of our lungs. The next type of connective tissue is cartilage, and it comes in three different types: hyaline, fibrocartilage, and elastic cartilage. Now, the word hyaline means glassy, which is to describe, under the microscope, hyaline cartilage kind of looks like stained glass if it's been stained well. And the other two are similar to that, but they're different in their composition. Fibrocartilage has a lot of collagen fibers in it, and gives it more strength. And then elastic cartilage has elastic fibers in it, giving it the ability to bend and snap back. Now, elastic cartilage and elastic dense connective tissue share the elastic properties given to it by elastic fibers, but they are different types of tissue. What defines cartilage is that there's an additional thing found in the ground substance, which is a large molecular structure which contains chondroitin sulfate. And that large molecule I always kind of think of it as kind of a fuzzy or hairy molecule, and so multiple molecules will kind of mesh together, kind of like a pet fur collecting in the corner of your apartment. And that gives cartilage a very rubbery consistency. And then the fibrocartilage has more strength to it. Elastic cartilage has a bit more bend to it and snap. So they're similar. This figure shows the three types of cartilage side by side and points out some of their similarities and differences. They're not stained exactly the same. Actually, probably if you stained fibrocartilage and elastic cartilage like we stain hyaline cartilage, it'd be pretty much indistinguishable. But the fibrocartilage image here has been stained to highlight the collagen bundles, and the elastic cartilage has been stained to highlight the elastic protein. Now, a good way to talk about their function is also to consider where they're found. So let me talk about that for a second. And I'm going to start with elastic cartilage. There are two places where we find elastic cartilage in this course. One is in the external ear. You can feel the ear, except for the lobe. Has a very rubbery consistency, and you can bend it and misshape it all you want, and it'll go back to its original shape, which is important because the shape of your ear helps with an aspect of auditory function, which is sound localization. Then we also find elastic cartilage in the epiglottis, which is a flap of tissue that covers the lower respiratory tract when we swallow. And the elastic cartilage makes it capable of being pulled down to cover that tube, and then it pops right back open to keep the airway open when we're not swallowing. Now, it might seem like we don't swallow that much in our lives, but really we're swallowing constantly. The mucus from our nasal cavity and saliva from our oral cavity is constantly moving down, and we want to be sure that that goes down the esophagus and not the trachea, so the epiglottis covers it up, along with the food that we swallow that we definitely don't want to go down the, quote, wrong pipe. Fibrocartilage is found in really three places that we're concerned with, which is the intervertebral discs, the pad of cartilage in between the vertebral bones and the vertebral column, the pubic symphysis, which is another pad of cartilage where the two pelvic bones come together in front at the pubic arch. And then in the knee, there are structures called menisci. There's the medial and lateral menisci, which are also fibrocartilage. Now, all three of those locations are important because they bear a good bit of our body weight. So the weight of our torso sort of bears down on our vertebral column and even comes down to bear on the point where the pelvic bones meet in the front. And then all of our body weight's bearing down on our knees, and so there needs to be some padding there. The important thing about fibrocartilage is that they all help to absorb that compressive weight by having the collagen fibers there, giving it some strength. If you look at the center of a fibrocartilage pad, it's going to look very, very much like hyaline cartilage. It's at the edges where there's a high incidence of fibrous proteins kind of wrapped around the cartilage cells, the chondrocytes. And then hyaline cartilage is found everywhere else. So if it's not in the external ear or the epiglottis or the intervertebral discs or the pubic symphysis or the menisci in the knee, other than those five places, all cartilage that we're concerned with in this course is hyaline cartilage, which is your basic kind of cartilage. Now notice in the figure for the hyaline cartilage they're highlighting chondrocytes in lacunae. Now, actually, we would find this in all of the different cartilages, but chondrocytes, or cartilage cells, are found within a little gap in the cartilage matrix, or the cartilage ground substance, called a lacuna. The word lacuna means a little lake, and the idea is when we look at it, there's a little lake that the cell's floating in. What's actually happened is the chondrocytes have been instrumental in making that matrix, and they've surrounded themselves with the matrix. So the cell was there first, and the matrix came in second. And the lacuna is just a representation of where the matrix isn't found because the cell was already there. Now we have bone, which we consider having two different types, compact bone and spongy bone. Now, bone tissue is kind of all similar. What makes these two different types special is the fact that compact bone is more organized. And we'll see that in the micrograph in a second. Now, bone tissue is a little bit different from the other connective tissues in that some of it actually doesn't come directly from mesenchyme. Instead, mesenchyme becomes cartilage, and cartilage calcifies into bone. There are some bones that directly develop out of mesenchyme, but the majority of them actually develop out of cartilage during fetal development. When we look at the micrograph of compact bone, we can see that there's a recurring pattern of these sort of circular structures with dark holes in the middle. Those are called osteons. The dark hole in the center is actually a central canal that's going to have blood vessels in it. And then it's surrounded by rings and rings of bone matrix, which is what we call the ground substance of bone tissue. The rings are separated by little flecks of black stained stuff, which are the osteocytes. And the osteon, the whole structure, is kind of a column structure which gives bone its strength. And architecturally, columns are very strong to hold weight in a vertical position. And so we see that in compact bone. Compact bone, however, is a continuous field of bone matrix. So where two osteons come together, since they're somewhat circular in shape, they're going to be little triangles or gaps of area, and that's filled in with more bone matrix, which just isn't organized into osteons. Where we see the black stain going from the center of one osteon to the center of another osteon, such as on the left side of this micrograph, that's where blood vessels from one osteon to the other are connected to each other. Bone is living tissue, and so it has to have a blood supply. And the matrix forms around the blood vessels and around the osteocytes and establishes the supply of blood to the tissue so that it stays living. When we look at bones in the lab, we're looking at the remnants of it. There's no living tissue left. It's all the bone matrix. But you can see the solidness of compact bone as opposed to the spongy look of spongy bone, which we'll talk about next. In the micrograph of the spongy bone tissue, what we see here is actually two tissues. Spongy bone tissue is called spongy because it has a lot of gaps and holes in it. The actual bone tissue in this micrograph is really just the pinkish, almost reddish color, and then filling in the spaces between the lighter staining part is either mesenchyme or bone marrow. It depends on exactly what stage this bone image has been taken. But that's what we'd see in mature bone. There'd be red bone marrow filling in the gaps within spongy bone. The chondrocytes are still in layers of bone matrix here. This isn't a high enough power to really see it, but it is true. And then on the outside edge of the bone here, which would be true on the outside edge of compact bone, there are osteoblasts, which are stem cell kind of cells, which are important for making bone matrix. And there will be a type of cell called an osteoclast, which is a counterpoint to an osteoblast. It helps to take bone matrix apart. We'll talk about all of this stuff in the bone chapter coming up soon. Now, for liquid connective tissues, the book presents blood and lymph as connective tissues. I don't really agree with lymph being considered a liquid connective tissue. It's really liquid from other sources, and the cells that make it up are really blood cells for the most part, white blood cells. It doesn't have its own real ground substance in the sense of fibrous proteins associated with it. So I don't really think it qualifies as a tissue in its own right. It's a bodily fluid. It's just not really a tissue. This micrograph is of what we would call a blood smear. Being a liquid tissue, we can't section it into solid tissue like we can with others. Instead, a drop of blood is put on a microscope slide, and it's smeared to the point where the cells are a single layer thick. The majority of cells that we see here are erythrocytes, or red blood cells. The lymphocyte is a type of white blood cell. And the number of white blood cells compared to red blood cells is pretty significant. There's probably 1 white blood cell for every 500 red blood cells, which isn't even what the ratio on this slide suggests. Also, there's a type of cell or cell fragment called a platelet, which is not labeled in this picture. If there is one here, it's the little dot that you see a few red blood cells below the white blood cell. It's not labeled because it's possibly not a platelet. At this magnification, it's hard to say for certain. It could be a speck, a piece of dirt, or something like that. But there are platelets there too, which are responsible for blood clotting. Now, blood is different from the other connective tissues because it does not contain collagen. All of the other connective tissues do, but collagen's not present. Instead, the fibrous proteins that we associate with blood is a tissue called fibrinogen, which is part of blood clotting. There are other plasma proteins that are more globular in structure than fibrous, but they make up the ground substance. If blood were exposed to collagen, that would actually cause blood clotting, and it's a useful little signal so that blood knows when it's no longer in the circulatory system. Consider this question. When you feel that you've reached an answer, hit the next button to go into the next slide, which will reveal the correct answer. So again, collagen's not found in blood. It's found in all the rest. And if blood is exposed to some other connective tissue, it's going to initiate clotting. The type of connective tissue that blood's most likely to be exposed to first is areolar connective tissue, as it makes up the basement membrane below any epithelium, such as the epithelium lining blood vessels. And so it's usually the first thing that blood runs into, and it has collagen in it, which stimulates blood clotting.